Okay, so welcome everyone, and thank you for joining us today. So before we begin, just to let you know that this session will be recorded. And now I will introduce our speakers. Um, we have Dorothea, and she's a bioinformatics intern at the Cyprus Institute of Neurology and Genetics and a data science student. Dorothea has previously worked as a data analyst in the gaming industry and as a analyst in consulting, and also now she applies her strong analytical and data-driven skills to biomedical research, with particular interest in machine learning and open science. And also we have Yelyzaveta, if I pronounced it correctly. She's also a bioinformatics intern at the Cyprus Institute of Neurology and Genetics, and an incoming PhD student at King's College London. She also has a hands-on experience in building computational pipelines, and she's passionate about developing tools to advance personalized medicine, and this is particularly in disease diagnosis and progression prediction. And today we are here to explore a fascinating and a rapidly evolving area in biomedical research, the role of microRNAs as key regulators across many biological processes. So uh, the floor is yours. Thank you.
Thank you, Valeria. Let me share my screen. All good? Great. So thank you for joining us today. For the past few months, me and Lisa have been working on this project at the Cyprus Institute of Neurology and Genetics, Bioinformatics Department. And today we're going to be taking you through the Altamir V1, which is a Python-powered database and analytics of miRNA disease associations.
So let's start with background. What are microRNAs? Well, our DNA is made up of genes which provide the instructions on how our cells function. In this context, microRNAs are tiny molecules that act as regulators, helping control which genes are switched on and which genes are switched off and kept off. So this regulation is highly sensitive. When something in this process goes wrong, it can contribute to a range of serious diseases, including various types of cancers, neurodegenerative diseases, as well as autoimmune disorders. So by studying which microRNAs are dysregulated in a disease, we can A, diagnose conditions earlier, B, predict how diseases may progress, and finally, develop new therapies that specifically target these faulty regulatory signals.
Thank you. Currently, miRNA-related data is spread across numerous independent databases. This information is fragmented, often inconsistent in terms of identifiers, data formats, and scoring metrics. This lack of uniformity makes systematic analysis difficult. Moreover, there's still no integrated resource that brings together all of this diverse information, such as miRNA disease associations, drug interactions, gene targets, and genomic variants, into one accessible and standardized framework. Our aim is to address this gap. Specifically, we aim to create a unified miRNA database by combining data from multiple high-quality, publicly available sources, standardize the information on miRNA disease links, drug interactions, gene targets, and variant associations, and finally, perform systematic analysis to identify patterns and common features among miRNAs that are associated with particular diseases. This integrated approach will help us better understand the role of miRNAs in disease mechanisms and potentially uncover new diagnostic or therapeutic opportunities. For the implementation, we relied on a combination of Python libraries for data handling, analytics, and visualization. For the unified miRNA database, we mainly used pandas for loading the datasets, SQLite as the database backend, and Streamlit for building the interactive SQL query interface. Data normalization steps like regex cleaning and ontology mapping ensured consistency across the datasets. For our upcoming disease similarity analysis app, we used a wider range of tools. In analytics, we apply a t-SNE for dimensionality reduction and a variety of clustering algorithms such as k-means, BERT, and Gaussian mixtures to identify groups of related diseases. We also built disease similarity networks using NetworkX and PyVis. Finally, for visualization, we combined Plotly for interactive scatter plots, Seaborn and Matplotlib for heat maps, and PyVis for exploring similarity networks interactively.
Okay, so we initially examined 25 databases as potential sources for building our own integrated database. From these, we selected the most suitable ones based on a set of criteria. So first of all, we looked at data relevance and uniqueness. We prioritized data sets that offered biologically meaningful and non-redundant information related to human microRNAs. Then we also looked at credibility and established use. So we chose databases that are widely cited, well-maintained, and curated by the scientific community. Also, we chose sources that had easy access. We prioritized datasets that were available for manual download and didn't require API access license or specialized software tools to access. Also, we looked at recency of updates. We only included resources that have been updated within the last 10 years to ensure data is still relevant. And we also prioritized sources that had clarity in their documentation. And we prioritized, well, we only had focused on human-specific data, so human microRNAs. So after this filtering process, we ended up selecting 11 databases out of the 25 we had initially. Many of these databases contained multiple data sets internally. So in total, we extracted and curated 25 individual data sets, which we then used to build our unified database. So the type of data collected could be summed up into 6 categories. Where the most relevant for our analysis platform was reference and foundational microRNA data, as well as microRNA expression and disease associations.
First, we harmonized the column names across all the datasets. Since the original sources all used different naming conventions, we mapped all column headers to a common schema, which ensured consistency and facilitated automated processing later on. Secondly, we defined strict formatting rules for each type of data field, whether it's Boolean, string, or numeric, and applied validation tests to make sure that all entries conform to these formats. This step helped us catch and fix errors or inconsistencies early in the pipeline. Third, we addressed the common source of ambiguity in miRNA-related datasets, the reference to genes. Many sources don't clearly distinguish between different types of gene associations, target genes related by miRNAs, host genes where miRNAs are embedded, and transcription factors that may regulate miRNA expression. So we made sure to explicitly label and separate these categories to avoid misinterpretation and enable more accurate downstream analysis.
A major hurdle in data standardization for us was dealing with disease and drug terms. So most microRNA disease and microRNA drug associations are extracted from scientific literature and clinical trial reports using text mining algorithms. Consequently, inconsistencies such as spelling errors, disease synonyms, and in the case of drugs, multiple compound names or multiple registered trade names introduced significant redundancies that had to be resolved through ontology mapping. So for diseases, we applied a 2-step strategy. So first of all, we proceeded with ontology-based mapping, where we assigned standardized disease identifiers to input disease terms using the NCBO bioportal annotator, which allows you to choose different ontologies for the ontology mapping. We specifically chose disease ontology and also medical subheadings, which are the most common and diverse, comprehensive ones. So as an example, here's the case with kind of how input and output may look for Alzheimer's disease. As you can see, we allowed both complete matches and component matches. So in the case where, like in Alzheimer's, there is the main type, which is Alzheimer's disease. There's also subtypes or more specific categorizations of the disease, such as late onset Alzheimer's and early onset, so that these could also be categorized as with both component and complete annotations. Yeah. So if the ontology mapping didn't work and some terms did get skipped over, we proceeded with manual curation, where we would manually categorize these diseases, which was supplemented with an in-house dictionary that we made that would handle these rare conditions. Overall, we were able to achieve about 96% annotation success on a data set of 2,855 unique disease terms. In the case of drug identifier assignment, we assigned chemical IDs as well as SMILES using PubChem identifier exchange service. So here, for obvious reasons, we only allowed complete matches because if a compound and a drug is a drug, you can't just use like a component of its chemical name. It wouldn't make sense. It could be a completely different drug. But anyway, here's an example of how an input and an output would look. So in the original tab, you can see the names of input drugs. And these came in all sorts of colors. It could be like a code. It could be just a drug trial name reference, or it could be like a licensed pharmaceutical name. And by inputting these through PubChem identifier exchange, we were able to derive chemical IDs, which could have, where a compound could have multiple chemical IDs, as well as a SMILES notation, which is just the chemical formula of the compound, where there would just be only a drug could only have one chemical compound. Yes. So we achieved an ontology mapping rate of about 87% for the drugs.
So we have a big diagram here that demonstrates our database schema, which organized all of our information about microRNA and their associated diseases, drugs, etc. So at the core, we have tables for microRNA sequences and annotations capturing their identifiers, genomic locations, and basic features. And around that core, we have set three main themes. We have disease links, which are multiple tables connecting microRNAs to different disease databases, including cancer studies, expression data, prognostic biomarkers. Secondly, we have drug interactions, which are predicted and curated data showing how drugs could influence microRNAs and vice versa. And third, we have genetic variation, which are SNP tables tracking how genetic changes may alter microRNA function and disease risk. Finally, we also have regulatory relationship tables, which map transcription factors, lncRNAs, pseudogenes, small RNAs, and epigenetic factors that interact with microRNAs. Altogether, this schema integrates sequence, disease, drug, SNP, and regulatory information into a single framework. Let's see how this database works in real time with our demo. Now, for a quick video demo of our SQL Query Explorer app, which we built in Streamlit, let's start with running the sample query to get a feel for the database structure. What this does is it pulls out the names of all of the tables in our unified database. And as you can see, we get a set of tables covering different areas, from core microRNA information, disease associations, drug interactions, SNPs, transcription factors, and more. Opening the sidebar on the left, we see a section that is dedicated to all of our available tables, and we're able to click on these tables to see their respective columns, making querying easier for someone who may not be previously familiar with these datasets. So the sidebar acts as a built-in schema browser. Now, let's take it a step further and run a more interesting query that actually links datasets together. I'll join the HMDD disease association table with the drug miRNA interaction table. What's happening here is we're taking the disease HMDD table, which contains experimentally validated microRNA disease links. Then we're joining it with a drug miRNA MIRMOL HSA table, which records drug microRNA interactions. The join is done on the shared standardized column miRNA ID. The output gives us a table with three columns: the microRNA ID, the disease it's linked to, and the drug associated. We're able to search for results for specific diseases, for example, cancer, which exhibits over 1,000 results. And the user can download the results as a CSV, which could be useful for further analysis and plotting graphs and diagrams.
So now that we had all of our data curated and ready for exploitation, we wanted to create a tool to demonstrate how the data we collected can be integrated to draw new insights about how similar are diseases to each other based on their microRNA profiles. So here's what we did. First of all, we measured how much two diseases overlap in terms of dysregulated microRNAs, which was done first by constructing a disease microRNA matrix, and then by calculating the Jaccard similarity between disease pairs. Then we turned those similarities into distances and projected them onto a map using t-SNE. So we can actually see the disease clusters. Next, we applied clustering algorithms, such as k-means, to group diseases that share similar microRNA signatures. So the result is, through this, that we can now identify clusters of diseases that may share molecular mechanisms and even potential comorbidities. So finally, users can explore results visually through interactive heat maps and a network graph on our Streamlit app, which can be accessed via this link. And in the next slide, we have a short video demonstrating how it works.
Let's go through our disease similarity analysis app, which allows users to explore how diseases relate to each other based on shared microRNA profiles. Our landing tab describes the concept. We build a disease similarity matrix using the Jaccard index, where each pair of diseases is compared by how many microRNAs they share. It explains how we use clustering, dimensionality reduction, and network visualization to explore those similarity relationships. We also have embedded links for keywords if we use any term that the user may be unfamiliar with. Let's move to the clustering view tab. Here, we can see the diseases represented in a lower dimensional space, so we can visually inspect which diseases cluster together, meaning that those diseases have similar microRNA profiles. This clustering can be adjusted to the number of clusters and the clustering method. Available, we have the k-means, BERK, hierarchical, Gaussian mixture, mean shift, and the affinity propagation. This app computes t-SNE on the distance matrix, then it assigns cluster labels and shows a scatter plot with diseases colored by cluster. If you hover over a point, you can see the disease label, and we can zoom into a particular area by selecting it. You can also select a specific disease you have in mind and see its cluster's other members and the top and most similar diseases. Below this, we have a heat map, where we can explore the similarity of a cluster of diseases and how similar they are to one another based on the Jaccard index and the microRNA profiles. Finally, we have the Disease Similarity Network tab. This lets us see diseases as nodes in a network, with edges showing disease pairs whose similarity exceeds the threshold. Again, we have some controls to play around with, and we're even able to filter diseases by keyword, for example, lymphoma or cancer. Once the user has set the threshold and selected diseases, they get an interactive network. Nodes are diseases, edges connect diseases with similarity above the threshold. Edge weights may reflect how high the similarity is. You can zoom, pan, and sometimes view details on hover. For example, if we select this line, we can see the diseases associated and their similarity index. Finally, we have our conclusion. To wrap up, here's what we built. We created a unified miRNA knowledge base with an analytics layer, and on top of that, we've developed two interactive Streamlit apps, the Query Explorer, which lets users search and filter across datasets, and the Disease Similarity Profiler, which helps uncover relationships between diseases based on shared microRNA profiles. Specifically in terms of scale, we integrated 11 public sources, captured over 4,500 verified microRNAs, and mapped them to over 2,000 diseases. We also pulled together more than 10 relationship types, including targets, ncRNAs, SNPs, epigenetics, and drugs, resulting in over 109,000 unique miRNA disease associations as of April 2025.
So why does what we did matter? So our data overcomes the data wrangling aspect of dealing with microRNA data coming from different sources. So it is pretty much ready for an integrative analysis for scientists who want to delve into microRNAs, human microRNAs, in the context of human biology. Also, since in our data, we have multiple interlinked layers of microRNA relationships. So as Dorothea has mentioned, we have microRNA like targets, both protein and gene, their relationships with non-coding RNAs, variants with drug molecules, etc. This enables scientists to form cross-layer hypotheses as well as explore them. So for example, building on what we've done with microRNA disease profiling, what could be done with our data is like we could build potentially a very crude diagnostics tool. And what I mean by that, we could take like patient uh, dysregulated microRNA profiles, just like what kind of microRNAs are dysregulated in a patient's body, and use our data to see how the patient profiles compare to existing disease profiles in order for potential diagnosis, as well as also by looking at the microRNA drug relationships, we could also potentially predict actionable drugs in those specific cases based off microRNA dysregulation profiles in patients, as an example. So overall, Ultimir not only enhances existing analyses through curated and accessible data, but also offers a robust foundation for developing novel computational approaches and methods to explore microRNA function and significance in biomedical research.
That's all from us today. Thank you for joining us.
Thank you. Thank you very much. That was a very interesting talk. I wrote a couple of questions. Yeah, I mean, I don't have any relation to microRNA, so maybe my questions are, I don't know, very simple or um, not very good questions. But yeah, I'll ask because it was a really nice talk, and thank you very much for that. So the first one, these databases that you started with 25, but then you selected 11 of them. Are all these databases like contain miRNAs for only diseases, or is it the microRNAs are not only for diseases and they have like good quality ones? So how does this work?
So microRNAs they exist in all of us. If you have microRNAs, that doesn't mean that you're like going to have some sort of disease.
Okay.
It's just in diseases, oftentimes, a disease is when a biological process in a person's body is dysregulated. And biological processes are controlled by genes, and which genes
Mm-hmm.
are like active and which are not. So microRNAs, they kind of like regulate whether which genes are active and which genes are not.
Okay.
So there is a certain baseline of kind of like how many microRNAs and in what concentrations a person has to be healthy, right? So when this balance gets shifted into like any direction and becomes dysregulated, that could lead to dysregulation of biological processes, like in cancer, for example, that is um, cell renewal, which leads to tumors. When that process becomes dysregulated, we get ill. So, in that sense, by being able to see and study microRNAs, like their functions exactly in the human body and how like different, like whether there's too much or too little, how that affects us, that could help us draw important kind of like conclusions and insights about diseases and how they progress, etc. I hope that answered the question.
Yeah, so how do you access this? Is it like inside the human body and you have to like use a needle or something to take it out and check it?
For diagnostic purposes, normally it's you take some kind of like body fluid. In most
Okay.
cases, it's blood,
Okay.
sometimes spinal fluid. It's like when you go and get blood work done.
Okay. Yeah, yeah, yeah. Cool. Um, the other question that I wanted to ask is about just the databases. Were they all having the same type of data, or you had to exclude some data from each since they weren't very related? Did they all collect the same type of features, or you had to select like from those 11 databases just 5 features from each that were similar among them?
So our project was from the get-go more centered in terms of kind of like microRNAs and diseases, as you
Okay.
understand.
Yeah.
There are many different types of databases dealing with microRNAs in different contexts. For example, there's like species-dependent databases, like there could be like ones for like rabbits and different types
Mm,
of like rats too, like for
Mhm.
that kind of research. We focused on human ones. That was like the first kind of like big
Yeah.
criteria. Then we prioritized the ones which looked specifically into kind of like microRNA occurrence in diseases and also drugs, because those are obviously very connected. Um, but also in order to like build a comprehensive database, because everything in biology is interconnected.
okay.
We chose also other databases that dealt with not diseases, but for example, like the type of targets microRNAs, like where they bind, for example, like what genes they bind to, like which genes they control.
Okay.
Like not explicitly like diseases, or for example, which other small like molecules in the human body they interact with. And like that could all be linked to disease context, but that would require more elaborate study in a sense in order to link that all to diseases, if that makes sense. So
Okay.
we just collected a lot of different
Mm-hmm.
sources, but for our like analysis bit, we only used like 2 categories. So we used about like 7 or 8 data sets out of the 24, if that makes sense, for the
Okay.
profiling bit.
Okay. Cool. And in terms of that, it's a more technical question, I think.
Mhm.
Why Jaccard similarity and not like Euclidean or something else? I mean, I'm not sure if that.
No, 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 that's a good
Oh,
question. So we chose Jaccard index. So our data, when we were
mhm.
doing the analysis, right? Um, we didn't have any information, like we didn't have any weights attached to this
Okay.
microRNA dysregulation. So it was just either microRNA in body is dysregulated or normal, like yes or no. It was like binary. Is that the
Binary,
right word?
yes. Yeah.
Yes. So for that reason, we chose Jaccard since it measures only overlap or union. Also, it's pretty robust on different size lists. It had like a 0 to 1 score, which we thought was very good for like heat map. It was easy for us to interpret. And also, it was just easy in terms of running it. It didn't take that much time, that much computational power. Of course, in the future, like to be if we had more time to work on it, in some data sets, they would talk about like they would give weights to kind of how which microRNAs are dysregulated more or less in a disease, right? So with these weights, we would probably use a different uh, index or metric. I don't know, maybe like we would add Fisher p-values, or maybe we would do like a weight Jaccard index, but we didn't really get to think about that that much because we
Okay.
ran out of time a little bit.
Okay. So basically, a disease has like multiple microRNAs, and you can put weights on it based on the disease. Did I
So
understand? Kind of.
imagine like if you take a blood sample
Mm-hmm.
from a person and you do like this microRNA profiling. You look which microRNAs are present there and at what concentrations, at
Okay.
what
Yeah.
quantities. There is a certain baseline which is like healthy for
Mm-hmm.
each microRNA. And you look how much each microRNA deviates from that healthy point.
Okay, I
So
see.
like
Yeah.
that's like a value. And based on that, you can attach weights to different microRNAs in different situations, if that makes sense.
Mm-hmm.
But unfortunately, we wanted to do that initially, but we couldn't find a lot of data sets that had these like values, like quantified deviations, if you know what I mean, like not
Yeah.
just dysregulation. Plus, like in biology and research, it wouldn't be very like statistically significant comparing them between each other because the way these metrics, these weights are derived is different in every research group. So it'll
Okay,
be more
yeah.
complicated, but it could be done probably in the future.
Okay. Okay. Cool. And my last question, can someone find your database online? Uh, and can they access it and make like more analysis that they would like to?
Yes, we have links to both of our databases in the presentation. And if
Okay.
someone wants to take the code and alter it for their own data sets or add more onto it, if they would just like to do that, that's available on my GitHub.
Okay. Great. That was a very, very interesting talk. It wasn't something that I normally know about, but it was, yeah, really nice, very interesting to learn about these microRNAs. Great. Thank you very much for your time and for your talk.
Thank you.
And yeah, I hope to see you and meet you sometime as
Yes,
well.
That would be great.
Thank you very much. Thank you.
See you. Bye bye.
Bye.